Welcome you all. Today we are going to discuss something about our first card, that is the introduction of biochemistry. We have completed this. Second is the acid, base, pH, pK, buffer and Henderson hasselbalch equation. These are the contents of our first item. First of all, we want to talk something about the acid and base. Why these acid and base are in the biochemistry? Because when the chemists are trying to discover the molecular nature or the chemical nature of the body, they are finding different chemicals that are functioning within the body, and the physicians or the people who are dealing with the human body, they are trying to take the advantage from the invention of this chemist. So the interaction between this chemist and the biologist is the biochemistry, and in this subject there are so many molecules. These molecules interact with each other and body has a solution. And we know that 60% of our body is composed of water, and the molecules that are dissolved in the water, they can behave sometimes as acid and sometimes as base. So for this region we have to learn the acid, base, pH, pK, buffer, all the things. I think that you have learned it in your intermediate lesson, and you have the strong foundation about these things. Today I want to just introduce you with this topic and the biology. In everyday life, we take our food, we take oxygen from the air, and there are some metabolism inside our cells. Our cell remains in a state of aqueous solution. That means there are more water within the cell, and the cell lies within a aqueous environment, within a water. The water that remains within the cell, that is known as the intracellular fluid or intracellular water, and the water that are outside the cell, that is the extracellular water. So during the metabolism, the end product of metabolism, what is produced inside the cell, that is transmitted outside the cell with the time being. And during the metabolism, we know that there are more and more production of acid. So this acid, production of acid inside the cell, that will transmit to the extracellular fluid, and production of acid, further acid, that will also transmit to the extracellular fluid, and production of this type of acid will accumulate within the body if there is no system to eliminate this thing. So our body has a system that can eliminate the end product of the metabolism. Usually the end product of metabolisms are acid. Body has different systems to eliminate and neutralize these things. When we take oxygen from the air and it enters into our circulation and to the cell, then the metabolic reaction, after the metabolic reaction, production of carbon dioxide, this carbon dioxide finally eliminated through the lungs by exhalation. This is one way of eliminating the metabolic end product. Another way of eliminating the metabolic end product through the kidney. With the dissolving in the water, the waste products are transmitted to the kidney and it is excreted through urine from our body. These are the eliminating systems. And where after production of acid within the cell, they migrate to the extracellular fluid. In the extracellular fluid, there are some systems that can neutralize the acid. The solution that has the capacity to resist the change of its character. Character means acidic character or the basic character. That is known as the buffer. So our body has different buffer systems. For the learning of the buffer, we have to learn something about the scale through which we can measure the acid, through which we can measure the base. In your intermediate learning, you have learned that the scale for measuring the acid base of a solution is the pH. That means power of hydrogen ion. It is nothing but the negative logarithm of the concentration of the hydrogen ion in the solution. The neutral method like water has the pH value of 7. The pH value more than 7 is the basic or alkali, and pH value less than 7 is the acidic. This is the fundamental thing of the pH. When we see that there is a solution and this solution has a pH about 5, so it is an acid solution, acidic solution. And our body, our extracellular fluid has a pH about 7.4. About 7.4. It has a range about 7 to 7.8, like that. So our body has a tendency to maintain a constant pH of its extracellular fluid. During this time, there are more and more acid adding with this body fluid, but our pH value of our extracellular fluid remains constant using these buffer systems. The main buffer systems of our body are the bicarbonate, carbonic acid buffer, phosphate buffer, hemoglobin buffer, protein buffer. These are the buffer systems. So how do these buffer act and how these chemicals interact within the body? For this region, we have to understand something about the dissociation constant. Dissociation constant means that if we allow the solution or the chemical into the water, or how much portion of the chemical or the solution will be dissociate within the water. That is the dissociation constant. If someone has, say for example, if the water has the water is a molecule composed of hydrogen ion and the hydroxide ion. So in the water we have the concentration of hydrogen ion and a concentration of the hydroxyl ion and the concentration of water in the room temperature. So if we want to measure the dissociation constant of the water, we have to measure the concentration of the hydrogen ion, concentration of the hydroxyl ion and the concentration of the water itself. This is an equation. Now we can see the equation here. Here we see the dissociation constant of the water. Here is the concentration of hydrogen, concentration of the hydroxide ion and the concentration of water in the room temperature, and we can see that it is 1.8 into 10 to the power minus 16 millimole, 16 mole per liter. It is the dissociation constant of water. So every molecule has its dissociation constant, and this helps us to define how this molecule react in the water. And the negative value of this dissociation constant, negative 10 base negative logarithm of this dissociation constant is known as the pK. Here we see that pKa, it is the minus log of Ka. Ka is nothing but the dissociation constant. Here we see there is a reaction where there is a weak acid and its conjugate base, and here is the dissociation constant and the 10 base negative logarithm of this decision constant form this pKa. And henderson hasselbalch equation, he shows that the pH is equal to pKa plus log concentration of the conjugate base divided by the concentration of the acid. This conjugate base means our body has the weak acids in the metabolic end product. This weak acid divides into the hydrogen ion and the conjugate base. This is the conjugate base, and in the henderson hasselbalch equation we can see that the negative logarithm of this conjugate base divided by the concentration of the weak acid, we can measure the pKa of that material. This pKa help us how this molecule will react in the solution of a certain pH. Our body pH is 7.3 to 7.4. So we have to measure the pKa value of different acids. Here we can see some acids that are produced during our metabolism. From the carbon dioxide in the tricyclic acid cycle, the product is the carbonic acid, and lactic acid produced by aerobic glycolysis. Pyruvic acid is the anaerobic glycolysis. Citric acid from the TCA cycle, and the acetoacetic acid and beta hydroxybutyric acid. These are the acids from the fatty acid oxidation. And from the dietary source we have the ammonium and the phosphate. These are the acids within human body, and these acids has different pKa values. Here we can see that the pKa value of carbonic acid is 3.8 and pKa value of pyruvic acid is 2.3, and there are different pKa values of different acids. But here is the sulfuric acid. It is completely dissociated. That means it has a pKa value less than 1. That means minus pKa value. Seeing this pKa value, we can determine that how much this acid will dissolve in our body fluid. Our body fluid has the pH about 7, more than 7, and this phosphate has the pH about 7 and it has the pH about 9. But all the acids has the pH is less than our body fluid. So it will dissociate less at that pH. So this is the importance of pKa and Henderson-Hasselbalch equation. So thank you all.